Dr. Martis has given you a good idea about the differences in production. Now I'll tell you about the differences in technology, and you can see the machines. Both technologies have some similarities. At the beginning, we start with input DNA. As Dr. Martis said, that DNA can come from a variety of sources and can be from a single organism or from a mixture of organisms. Before starting, the input DNA is broken up into many short fragments of DNA. The first step of the sequencing process for both machines is to make the input DNA into a library and to amplify each fragment, making many copies of each piece of input DNA. This step doesn't involve bacteria like the old sequencing pipeline, just DNA polymerase. At the end, the sequencing reactions are detected by a camera observing a flash of light. In the middle, there are many differences. Let's start by talking about the Illumina technology. Illumina uses two machines called the cluster generator and the genome analyzer. If we have full-length genomic DNA, the first step in making a sequencing library is to break the DNA into smaller fragments. This is done in a nebulizer and library core. After the DNA is in smaller pieces, the ends are repaired to make them blunt-ended. Then we ligate or attach adapters to both ends of the small pieces of input DNA. The input DNA is represented in gray, and the adapters are represented in purple and pink. We add adapters so that we know the sequence at the end of the DNA strands, allowing us to design oligonucleotide primers. Remember that polymerases require primers to start copying a sequence. A flow cell is like a slide with 8 sample lanes etched on it and a cover slip positioned on top that's attached to a collection of tubes. Small volumes of liquid can be pumped through the tubes so that the liquid completely covers the surface of the slide. The machine pumps different amounts of different liquids through the flow cell to automate steps of a reaction. After the adapters are ligated to the ends of the input DNA, the DNA is denatured to make it single-stranded, and the sequencing library is loaded into the flow cell on the cluster generator. It is attached to the several sets of tubes that will deliver all of the reagents and enzymes needed for the reaction. The surface of the flow cell has a lawn of primers, short sequences that match the adapter sequences, shown here as short strings of pink and purple dots. When the single-stranded DNA is loaded on the machine, shown here in black, the adapters will form hydrogen bonds to primers with the matching sequences, pink adapters binding to pink primers and purple to purple. Polymerase is added and starts to make a copy of the template strand. The copied strand is shown in gray. Because the copied strand starts with a primer attached to the flow cell, that strand is bound to the surface of the flow cell. Then the temperature can be increased and the original strand is melted off, leaving a single strand bound to the flow cell surface, surrounded by a dense lawn of primers. There are millions of pieces of input DNA spread out over the surface of the flow cell, and each has a unique sequence. However, we can't yet sequence the DNA because there's only one copy, so we go through a process called bridge amplification, which is just another type of PCR to amplify the DNA so there are many copies sitting in the same place in the flow cell. The temperature is decreased so that the DNA stuck to the flow cell forms hydrogen bonds with the primer on the surface of the flow cell, shown here in purple, creating a bridge. Then a PCR reaction mixture is run over the flow cell. The temperature is increased and the polymerase makes a copy of the input DNA. The growing strand is seen in gray. We call this bridge amplification because the copying occurs in the arched region between the two adapter sequences. When the polymerase is finished, there is a double-stranded arch with one end of each strand attached to the surface of the flow cell. Then, just like any other PCR reaction, we heat the flow cell and the two strands denature or separate. In this case, one strand is attached to the surface of the flow cell by the pink primer, and the other strand is attached to the flow cell by the purple primer. We can then go through another temperature cycle. We lower the temperature so that each single-stranded piece of DNA anneals to a primer on the surface of the flow cell and forms a bridge. The temperature is increased to allow the polymerase to extend a copy of the DNA. The flow cell is heated, and we have 4 single-stranded copies of the input DNA. All copies are attached to the surface of the flow cell very close to one another. This can be continued for many cycles of amplification, leading to 1 million copies of single-stranded DNA attached to the surface of the flow cell in a cluster. Because we started the process by attaching many different pieces of input DNA spread out over the surface of the flow cell, we now have many individual clusters of the input DNA.
Why don't you do the PCRs in tubes? You can do a lot of PCR reactions in a microtiter plate.
This is a very different scale. Even in the best microtiter plates available, we could only do 384 PCR reactions at once. In the cluster generator, we can attach 200 million pieces of input DNA over the surface of the flow cell. Because the DNA is spread out and attached to different positions, a different PCR reaction occurs at each separate position. The amount of reagents we use is greatly decreased, and we end up with millions of clusters of DNA, each cluster having a particular sequence. Now, one of the strands is cleaved within the adapter sequence, and the reaction is heated to separate the strands. Because one strand is not attached to the flow cell, it floats away. Now, each cluster contains only single-stranded DNA of a given sequence. The sequencing primer is added, it hybridizes to the corresponding adapter sequence, and the clusters are ready for sequencing. The flow cell is removed from the cluster generator. For the next step, the flow cell is loaded into the genome analyzer. This machine also has reservoirs attached to a set of tubes to deliver the reagents needed during sequencing, such as buffers, DNA polymerase, and fluorescently labeled terminator nucleotides. Illumina and Sanger sequencing are alike. The bases are represented by a pentagon for the sugar backbone, a circle with a P for each phosphate group, and a rectangular box for the base A, T, G, or C. In the sequencing reaction, every base is labeled with a different fluorescently colored dye, represented by a star, and has a 3' terminator represented by a stop sign on the 3' carbon that keeps the reaction from proceeding beyond a single addition. Unlike Sanger sequencing, all of the nucleotide bases are labeled with fluorescent dye and have terminators. That means the polymerase adds only one labeled nucleotide to each strand of the sample DNA, then stops because of the 3' terminator. A laser excites the dye, which flashes with the color corresponding to the base. The color of the flash is detected by a camera.
That's a lot like the old sequencing method, but with the old method, the cycle couldn't continue after you added the dye terminators. How is this different?
The major difference is that after the first cycle, the dye and the terminator are both removed. Then another round of reaction mix can be run over the flow cell, and another base can be added and detected by the camera. Then the dye is removed along with the terminator block. The camera records the timing, color, and position of each light flash, so the sequence of the bases can be determined for each cluster. The flow cell is attached to tubes that deliver the reagents. It remains stationary with a laser pointing at it. The camera is positioned above the flow cell to capture images of flashes from all the clusters on the flow cell.
Reversible terminators sound like a good idea. Are there any problems with this sequencing method?
The removal of the dye and the terminator is difficult and not foolproof. If the dyes are not completely removed, then 2 colors will be read by the camera in the next cycle. If we fail to remove all of the terminators, then the signal strength will be decreased because fewer dye-labeled nucleotides will be incorporated in the next cycle. Because of these difficulties, the maximum read length of Illumina is about 100 bases, and there can be a small error rate of incorrect bases in the sequencing reads. Both the Genome Center and Illumina are working to increase the read length, but for many sequencing projects, this read length is sufficient.
What about Pairedend reads? Is that possible with Illumina?
Yes, There is a machine that can be attached to the genome analyzer called a Pairedend module. This acts like a small cluster generator to regenerate clusters for paired-end sequencing. After sequencing with one primer, the flow cell can be heated to separate the sequenced strand and then washed off. A few cycles of bridge amplification regenerate the other strand, and the first strand can be cleaved, denatured, and washed away. Then the sequencing can be repeated using a primer with a sequence that's complementary to the other adapter. Because the clusters remain in the same location, the computer can record the information knowing that this is the same fragment, and we get paired-end reads, sequence information that we know comes from the two ends of the same DNA fragment. This is very useful in assembling the sequence information. In an 8-day run for one flow cell, we observe about 200 million clusters with a read length of 75 to 100 bases each. The overall amount of sequence generated can be as high as 20 billion bases, which means we get 6x coverage of the human genome in 8 days. That means Illumina sequencers can obtain 1x coverage of the human genome in 1.2 days.
That's pretty amazing.
It is.